Hello and welcome to all my students. So today we are going to start with the next lecture of molecular basis of inheritance, in which we are going to cover about human genome project. So far you have seen the concept of operon in the last class, and yes, today we are going to see about human genome project. It is a very simple and easy small topic where you have to remember some few important points. So let's start with today's class. Beta, in human genome project, first of all, what this project is. This is a huge mega project which was launched in the year 1990 with the aim of sequencing the entire genes and the nucleotide present in the human DNA, with the aim of solving many genetical related disorders. We know that there are so many diseases like Parkinsonia, Alzheimer's, which now are curable. But before Human Genome Project, when we were not aware about the arrangement of genes in the human cell, then at that time, such dreadful diseases were incurable. Concept of gene therapy did not exist. But once, with the help of this project, when entire human genome, that is haploid set of chromosome, that is 3 into 10 to the power 9 base pairs, were decoded, their arrangement was decoded. So yes, the detection of the disorder also came into existence, and once the disorder reason is detected, automatically cure can also be discovered. So with the aim of solving number of disorders and other problems related to human genome, this project was launched in the year 1990. So first point which you are going to write is that human project was launched in the year 1990. Was launched in 1990 with the main aim of solving gene related problems, with the main aim of solving gene related problems. This is a mega project. Human Genome Project is referred to as a mega project. Now there are certain reasons associated why this project is referred to as mega project. Two reasons, rather basically three reasons. Number one, this project continued for 13 long years. So it started in the year 1990 and ended in the year 2003. So this project continued for 13 consecutive years. So it got completed in the year 2003. The aim was decoding the entire genome 3 into 10 to the power 9 base pair. Per base pair cost was $3. That means huge investment was required for human genome project, which was nearly $9 billion. So huge investment was required. Which was nearly $9 billion, which is absolutely a huge amount. Now, for decoding 3 into 10 to the power 9 base pairs of human genome, number of technicians, scientists, research scholars were also invested for a longer duration of 13 years. So, large number of technicians, scientists, were involved in this process. So because of so many reasons, it was extremely time consuming, energy consuming, manpower consuming, money consuming. So that is why Human Genome Project was considered as a mega project, right? It was launched by, basically this project was launched by two organizations. One was US Department of Energy and second, National Institute of Health. So US Department of Energy and NIH, National Institute of Health, these two organizations, they planned to launch this mega project, Human Genome Project. Later on, it was joined by Wellcome Trust of UK. So later on, Wellcome Trust of UK joined the project launched by US. Not only these two countries, then with the time France, Germany, China, Japan, all other countries also became the part of this Human Genome Project, because the aim was very good. Not only US required, not only UK required, but every individual country is facing with troubles related to genetic disorder. So every country required to know the sequence of the human so that they can give a good cure to their people, so that their country can be made genetic disorders free. So with that aim, number of countries later on joined the project initiated by US and continued by UK. It was a huge project that continued for 13 big years, and so much investment was given. Right? Next, so this is about the introduction portion. Now you have to check out the goals of HGP. Aims. Most important aim I have told you, but let's discuss goals of HGP in more details. Number one was obviously to decode the entire human genome sequence that consists of 3 into 10 to the power 9 base pair. To decode the entire human genome which had 3 into 10 to the power 9 base pairs. Second, now automatically, see, all the nucleotide sequence, they are not responsible for protein synthesis. Limited protein, limited number of nucleotide making the gene are responsible for protein synthesis. So next aim of this project was to know that which combinations of nucleotide are responsible for protein synthesis. That means their aim was to decode the genes located in the human genome. So second important aim was to know or to understand the gene nature of humans. That how many genes are there? So it's nearly 20 to 25,000 genes are there in total human genome. So what are those genes? What are their locations? How do they function? So this was also the aim of Human Genome Project, gene nature of human genome. Next, now beta, when you decode 3 into 10 to the power 9 means 3 billion base sequences, then information generated will be too huge. So the next goal was to store that information in the form of database. See, storing this information in the form of books is not possible. If suppose 3 billion letters has to be written, then thousands of book will be consumed. One whole library will has to be constructed just to keep information related to human genome, which is practically not possible, right? So now third important goal is information generated was very huge. Information generated after sequencing was huge, बहुत ज़्यादा थी. So next important goal or target was to store this information not in books. You cannot write the books. There has to be some software generated in which this information could be stored in computer. So that whole library has not to be created. So that store this information in databases and utilize these databases for different sectors, like for example forensic. Now when we know human genome, that is why only we are able to perform DNA fingerprinting. Yes or no? So use the information generated from it in different sectors, mainly in the field of forensics. So next important goal was use this information or pass the information of human genome in different sectors like forensic, medical, no, DNA fingerprinting, detecting the disorders, creating them medicine for curing. All these fields are the fields that require to know the human genome sequencing so that they can function efficiently. So all this led to the development of new branch of science called bioinformatics. Bioinformatics is that branch of science that deals with the storing of the information generated by sequencing. Now here it is human genome. It's not only that human genome has been sequenced, the other non-human genome project has also been launched, in which you are decoding maybe the genome of bacteria, any plant, any other animal. So all these decoded information need to be stored in databases, and that branch of science that deals with this database is known as bioinformatics. So this human genome project led to the development of a new branch of science called bioinformatics. Right? Oh, sorry. So these are the goals and aims of HGP. Now you have seen that human genome project is such a huge project. Its aim is so good, goals are so nice, and yes, this should be carried out so that we can help our fellow mates who are suffering from some of the other genetic disorders, isn't it? But how to do it? What scientists actually did to decode so many nucleotide sequences of the human genome? So now next is method of human genome project. So beta, there were two groups of scientists. Now automatically when such huge project has to be carried out, scientist has to do, technicians has to do. Now obviously there will be some conflicts between the scientists whenever there is a marriage at home. Marriage is such a small thing. In front of this human genome project, marriage is a very small affair. But then too, there are 4 family members, 2 going in one direction, 2 going in other directions. And it becomes very difficult to bring all the family members on the same page for just a small ritual of marriage. Now here is the matter of human genome project, where so much of money, time and etc. is invested. Automatically here also the number of scientists, they won't be on same page, right? Some scientists will say no, we are going to follow this method of gene sequencing. Some will say no, we are going to follow this method of gene sequencing. So similarly the same thing happened over here. There were 2 groups of scientists who believed in different methodologies for solving the human genome. The 2 most highlighted methodology were EST, called as expressed sequence tags, and second was sequence annotation. So there were 2 methodologies which were the most highlighting one. Some scientists supported EST and some scientists supported sequence annotation. EST means, now you know that beta, our whole DNA is not responsible for producing protein. All the nucleotides are not active. Only a part of the DNA is active, which is responsible for producing protein. That active portion is known as exon, and the remaining dead portion are known as intron. Now, some scientists said that what is the point of knowing the nucleotide sequence of interons? because interons, no matter even if they're existing in the DNA, they're never going to do anything. Even if some mutation occur in them, even some change occur in them, then it will be of no use. The phenotype will not be affected if any change occurs in the interon. So why to invest time? Why to invest money in simply knowing the sequences of interon which are of no use? Why not to do the same work in lesser time by consuming lesser money? Why to simply decode 3 into 10 to the power 9 base pairs, so much money, so much time to be given, even when half of the human genome is of no use? So why not to strictly go and study about those sequences which are actually going to affect the phenotype? That means why not to only decode the exons portion? Like this, time will be reduced, money will be reduced, manpower will be reduced. In lesser time, our work will be done. But some scientists says no. If we are doing it, we will do it completely. You never know when interons in future will become active and will affect the phenotype. So today, if we have started this project, let money go, let time go, let manpower go, but we will completely decode the human genome. We will decode all 3 into 10 to the power 9 nucleotides, even if half of the portion is of no use. So those scientists who believe only in decoding the exons, they believed in ESTs, and those scientists who believed in decoding the whole genome were supporting sequence annotation. So what are the two methodologies? ESTs. ESTs, expressed sequence tags. That means decoding the genome of only exons, not of interons. Sequence annotation says decoding the whole genome, including interons and exons. So automatically later on this technique was approved by all scientists कि चलो ठीक है भाई, we are doing it for the first time, and when something has to be done first time, it should be done completely. Why to take a shortcut? Okay, shortcut may be good for right now, but maybe in future this expressed sequence tag may become useless, and after 100 or 200 years, if new scientists come, then again they have to launch the same project. Then what is the use? If today we are doing it, let it do it complete. Why to do it half and leave it for future? So everybody came on the same page later on, and then sequence annotation was the technique adopted by the scientist for 13 years to completely decode the human genome, which consists of 3 into 10 to the power 9 base pairs. Clearly understood? Chalo, now we are going to see the steps. Now methodology, you know. Which methodology are we going to follow? Sequence annotation technique given by Frederick Sanger. So Sanger's sequence annotation technique was adopted for human genome project. First step is beta, isolate a cell. Any cell. Any diploid cell can be isolated. So first step towards human genome project was take out a cell. Now you have taken out a cell. Now after isolating the cell, take out all the DNA, all the genetic material from the nucleus of the cell. So second step is isolating the DNA. Now DNA is going to be huge. Now DNA in one dhaga, it cannot be studied. So next is breaking that DNA into smaller fragments with the help of the enzyme restriction endonuclease. So the third step is fragmentation of DNA. Beta, with the help of restriction endonuclease, also called as biological scissors. right? Next, now each fragment is represented by one sample, but after this, each fragment number should be increased so that chances of error could be reduced. So the next important step is that number of fragments should be increased. You all do this technique, you all do this in biotechnology chapter when you study in zoology, that using vectors, the number of the same DNA fragment can be increased. So here vectors were used. The vector like YAC, yeast artificial chromosome, or bacterial artificial chromosome, yak or BAC, as vector they can be used in order to clone the fragment. So number of fragments should be cloned or should be increased by using the vector for cloning. There are two vectors used called as BAC or YAC, bacterial artificial chromosome or yeast artificial chromosome. Now once the fragment cloning has been done, then yes, for decoding the sequence annotation technique is used, which was proposed by Frederick Sanger. So last step is, now after increasing the number, now use the sequence annotation technique proposed by Frederick Sanger. Technique by Frederick Sanger, called as Sanger's annotation technique for decoding the whole genome. Now once the decoding was done, some results, there were some outcomes after 13 years of this human genome project. Once the technique was completed, some features, some outcomes were generated. These are the 9 outcomes which are there on your board. Let's read them one by one. So first, say some of the salient observations of human genome project are: according to human genome project, the total number of nucleotide in human genome is 3164.7 million. So there are, in 3 into 10 to the power 9 base pairs, the number of nucleotide reported were 3164.7 million nucleotides. Second, the average gene consists of near about 3000 bases. Now one gene, you know, it is made up of number of nucleotide. So average one gene consists of 3000 bases. However, the largest gene is dystrophin, that is made up of 2.4 million bases, and the smallest gene is TDF gene, which only has 14 bases. The number of gene is highly variable. On average, 3000 nucleotides can be there in a gene, but the largest is reported in dystrophin and the smallest has been reported in TDF. I hope you're going to remember the 2 points. Next, the total number of active regions on DNA, that means the total number of genes, are estimated to be near about 30,000. Only 30,000 active regions are there on our human DNA, which can help to produce proteins, right? That means near about 99.9% of bases, they are of no use, and they are same in every individual. See, I and you, we are humans. Our 99.9% of genome is similar. Only 0.1 difference is there, and that 0.1 difference is actually making so much difference in our appearance. Next point, the functions are unknown. Genes, okay, near about 30,000 genes are there, but all genes functions are yet not discovered, right? Next point, less than 2% of the human genome codes for protein. That means this 30,000 gene makes less than 2% of the total genome, which is 3164.7 million. Now millions में nucleotide हैं, and only they are active. How much is active? Only less than 2% is active. Rest all are recessive. Rest all are present in the form of interons. Next. Now, in our human genome, major portion is repeated sequences. मतलब same sequences are again repeated. मतलब now when these people, when they were decoding all the nucleotides, they found that majority of the sequence on the DNA are repeating again and again. And these repeated sequence, they are interons. They do not code for protein. So major portion of our DNA is non-coding. Only less than 2% of the human genome is coding. Right? Yes or no? So what are repetitive sequences? These are the stretches of DNA that are repeated many times, maybe hundreds to thousand times. Next, they are having no function related to coding. They are simply interons. इनका कोई भी काम coding से related नहीं होता है. ठीक है? They are simply interons waste. Next point. Now, out of the 46 chromosomes of human, chromosome 1 has maximum number of gene and chromosome Y, which is the smallest, have the fewest number of gene. So the question comes, which chromosome has maximum number of gene? Answer will be 1. Which has least number of gene? Answer will be Y. Right? Yes or no? Understood? Now, you know our major genome is made up of repetitive sequence which are exactly same. Apart from that, nearly 1.4 million locations have been such where entire sequence is same except one nucleotide changes. So they cannot be called as repetitive sequence. Otherwise, if that one nucleotide would have been also similar, they would have been categorized as repetitive. But now since the sequences are only differing by one nucleotide, so they cannot be considered as repetitive. Such 1.4 million locations are called as single nucleotide polymorphism. सिर्फ एक न्यूक्लियोटाइड की वजह से दो sequences are not same. So SNP, scientists have identified nearly 1.4 million such locations where only single base is differing. Such locations are known as SNPs, single nucleotide polymorphism, or SNPs. The better way is to pronounce as SNPs. Correct? So these are the 9 important features related to human genome. Hope you have understood the concept of human genome, the goals of human genome, the methodology, terminologies related to human genome, steps and the features. So this much is there in your syllabus. Yes, again, homework is few questions. Homework number 1, homework number 2. Try to solve these questions. Very, very simple. Yes, with this, we come to the end of today's lecture. Again, a very simple, quick, crisp lecture. Only the last topic of this chapter is left, that is DNA fingerprinting, which we will do in our next class. Till then, thank you for watching me. Bye bye everyone. Take care.